cool that you're strong. I respect it, I do. But in the end, everybody breaks, bro. It's biology.
Hello. Just quick mic check. Just going for a little bit on biology to do with cell membranes. Did a poll, cell membranes won out. So going live on Wednesday. It's normally going to be Thursdays for bio, but yeah, I'm away tomorrow. So what are we gonna do? All right. So guys, got some pictures of cell membranes up here, and well, actually, I've got different cell organelles on the screen. The reason I'm doing like that, I've got my camera in the way. Yeah, so I've got different organelles you can see there on this picture because cell membranes are not just around the outside of biology and plant cells. Well, they're around all cells, bacteria cells as well, fungi. Everything has a cell membrane if you're made of a cell, and all life is made of cells. But they're also around organelles as well. So cell membranes are important for compartmentalization within a cell. So they separate the contents of organelles from the cytoplasm. So that's an important thing as well. So cool. Yeah, so we've got the phospholipid bilayer up here, top left. That's a model that was proposed in 1972, the fluid mosaic model. Lots of other different proposed models. I'm not going to go too much into the different theories and models that were around before, but that is the currently accepted one. So we're going to have a look at the cell membrane. So first off, just a quick reminder of this biological molecule because it plays a little bit of a role in the cell membrane. That's right. Yeah, we want to separate compartmentalization. Absolutely. We want to separate digestive enzymes from and other chemicals that we don't want just going around the cell. So yeah, if you want to get inside the chloroplast, you got to pass through the membrane. If you want entry into the mitochondria, you're going to have to get through the cell membrane. So there's some control over what substances go in and out. Of course, that's especially important if you're going to change the water potential. If you're just going to let anything come in, and if it's going to affect the water potential, it's a disaster. So, what's the problem if we let sugars or salts, polar molecules that are soluble, into an organelle? What would be the problem if glucose molecules could just diffuse into, say, a chloroplast or a mitochondria? What would that do to the water potential? Anyone got an idea? I'll come back to what this image is, but yes, osmotic damage. Thank you. Yeah, it would decrease the water potential, and you get an osmotic effect. Water moves by a passive process from a high water potential to a low. So if you had sugars and salts just willy-nilly going in and out of organelles, they would lower the water potential. In would come water, and you'd have loads of organelles blowing up and cell structures exploding. So we do need control over what substances go in and out of cells. This, by the way, someone put up in the chat, is a triglyceride, and of course we're all told fats are bad for us because, of course, they build up in our coronary arteries and other arteries and can cause cardiovascular disease or CHD, atherosclerosis, however you say it, plaques, fatty deposits, all that business. But of course, a lot of the lipids you need are in your cell membrane. So if you didn't have any lipids in your diet, you'd be in trouble. This is a triglyceride. What have we got? What have we got, folks? Who can tell me what the different components of this triglyceride are? Ba, ba, ba. Let's call that C. Well, A and C, bit weird. Okay, let's put B there. So what's A, B, and C? Yeah, that's you're absolutely right. That's Mathilde or Mathilde. A good point. Yeah, you don't want your organelles to shrivel up as well. So you don't want water leaving them as well by osmosis. You want to keep things compartmentalized. So we've got 3 fatty acids, which is part C. B, what kind of bond is B that holds those fatty acids to A? And we've got the glycerol, which is A. Thank you. And B is the ester bond. Well done. Yeah, absolutely. B's the ester bond, holds together the glycerol and the fatty acid. Super. Why am I talking about triglycerides? Because you're not going to find a lot of triglycerides. You find some lipids in the cell membrane, but you're not going to find lots because, of course, the main thing in the cell membrane is our lovely phospholipid, which we're going to come to. Now, I'm just going to look at some of the questions before I move on. Oh yeah, that's why when we do homogenization, someone mentioned homogenation or homogenization, however you say it, before you do centrifugation, and that when you're separating the cell into all the different organelles, you want an isotonic solution because you want to prevent the osmotic effect. You want to prevent organelles shriveling or exploding. Yeah, absolutely. Um, good. Someone said, can you explain the function of the Golgi apparatus or Golgi body complex, whatever people want to call it? The consistent word or constant word is Golgi, and the function of the Golgi apparatus, which you will find in eukaryotic cells, the Golgi apparatus, is to package and modify proteins. So that might be taking a protein. Now, it isn't where the protein is first folded, by the way. The protein comes off of the ribosome or the rough endoplasmic reticulum, and it will immediately start folding due to hydrogen bonding between the different R groups. So it will immediately go into like beta or alpha coils and helix kind of thing straight away. But that will happen inside the rough endoplasmic reticulum, the folding, or after it's just left the ribosome. It goes to the Golgi apparatus for a little bit more packaging. So you can imagine if you're going to be a hemoglobin molecule, you need to be formed in a certain way, different polypeptides twisted around each other. If you're going to be a glycoprotein, you're going to need a glycogen molecule attached. And then, of course, it arrives and leaves in vesicles. And that's simply all you have to know at the A level, is that it's for packaging or modifying, changing proteins. So you could be adding a lipid or a glycogen molecule. Right. It's kind of AQA, OCR, EdXL. It's just some basics. I'm not going to go through everything because I'm just doing an hour. So I'm not going to go through everything. But be aware you could be asked. I have questions from OCR, EdXL, and AQA. Some of these questions that I'm going to put up are from all of these ones. So look, figure 1 shows a fatty acid that contains 20 carbon atoms and 4 double bonds. Oh, we don't want to know that. Draw a box around the R group of the fatty acid. So this is an AQA question, but you would have to know this if you were studying OCR. And I have OCR questions as well on this 1 hour live thing I'm doing on cell membranes. So I know I haven't got to the cell membrane yet. Oh, nice. Good point. How can fatty substance block up arteries if they're outside the vessels? They actually build up inside the wall, in the wall of the artery, and then push through. And you get damage to the arteries from high blood pressure. So the endothelium wall inside the artery gets damaged from high blood pressure. So if you imagine the friction of red blood cells banging past it all day and all night for months, years. So generally, if you have high blood pressure for a long period of time, that's going to be the biggest risk factor towards cardiovascular disease. You're going to get fatty, and of course, certain fats are deposited into your blood as well. So if you have cholesterol called LDLs, low density lipoproteins, they're going to remove lipids from your liver and your cells and place them into your blood. So therefore, you can get fatty deposits. So it's really the type of fats you eat and the type of cholesterol you eat. You want to avoid the LDLs. Anywho, rather than banging on about all of this, yeah, everything except the COH. Thank you very much. Yeah. So the R group of the fatty acid is all of that. Well done. That's the R group of the fatty acid. The carboxylic group will always stay the same. It's this that can change length, and it's this that could have one double bond, no double bonds. It could be all saturated. This is a polyunsaturated with 4 double bonds, but it could have one and be monounsaturated or no double bonds. So that's the R group. Coolio. Right, let's move on quickly off of this question because I want to get onto the actual membrane. Describe 2 differences between the structure of the triglyceride shown in figure 2, this one here, which we just looked at, and a phospholipid. Very common question if you're studying EdXL, OCL or AQA, to compare and contrast a phospholipid and a triglyceride. So what are the main differences? The R group, yeah, so one secondo. So you're going to get a phosphate group in a phospholipid, which is not found on a triglyceride. So phospholipids have a phosphate head, and they have 2 fatty acid tails instead of 3. So there's really 2 main differences. I can't think of any more than 2. Phosphate group for the head and 2 fatty acid tails. So I can't really think, instead of 3 fatty acid tails. Okay, now if you were talking about similarities, can you name in the chat 3 similarities between this and this? What are 3 similarities? Nice, you've all got that right. You've all smashed that, so well done. Give you a quick boom for that. Boom! Yeah, that would be nice if I didn't delay the boom. Yeah, doing very good. Yeah, both contain ester bonds. Nice. Both contain a glycerol and at least 2 fatty acids. Yeah, so they both have fatty acids, both have glycerol, both have ester bonds between the glycerol and the fatty acid. So there's 3 similarities there. Nice, lovely jubbly. Right. Let's move on and look at an actual membrane. Someone asked me, can I explain why that is the R group? So what it is, this is your glycerol molecule when you're looking at fats. This is your fatty acid tail, and essentially your carboxylic acid group is bonded here, and your carboxylic acid group is here forming the ester bond, so will always be the same of that fatty acid tail. That carboxylic acid group will always be locked in here, but then the tail can be different lengths. So just to show you the tail bit, the R group is the bit that varies. So the R group, this one could be, what's that? 1, 2, 3, 4, 5, 6 carbons long, and it's saturated, no double bonds. And we could make this one 1, 2, 3, 4, 5, 6, 7, 8 carbons long, and we could whack a double bond in there. The double bond would actually change the shape of that. It'll cause a kink, but we won't go into all of that. So essentially the R groups can be different, but all of them, all fatty acids, will contain this carboxylic acid group. So the R group is the bit that can vary between different fatty acid tails. They could all 3 or 2 in the case of a phospholipid be identical, or they might not be identical. So they don't all have to be identical. Cool. So they have the same contain amino acids, glycerol and ester bonds. They don't contain amino acids. There's no amino acid in these 2, but there are ester bonds and there are glycerol. So the carboxylate group is where the ester bond is, so doesn't change. Absolutely, yes. The carboxylate group is the part which is going to form that ester bond. You know, when you do an alcohol, so although we draw a rectangle here, if you study chemistry, it's where your hydroxyl group of the alcohol, propan 1, 2, 3 onwards, glycerol, it's where each hydroxyl group is going to bond in a condensation reaction, losing water to form the carboxylate group. Yeah, that's going to link them together. Absolutely. Right, let's zing to the membrane. Oh no, here's our phospholipid. So we can also draw the phospholipid like this. It looks like a sperm with 2 tails. These are the tails and this is the head. What do we know about the heads? What do you know about the heads here, guys? There's some terms you need to know for the heads and the tails. Lovely, lovely. Hydrophilic. So I'm not going to write everything down because when I start writing, it all goes a bit pear-shaped. See? Hydrophilic head. What does phile or philic mean? It means love. So a bibliophile is someone who loves books. Hydrophilic head. hydro really stands for water. It's kind of like, yeah, so it loves water anyway. And the tails are hydrophobic and they don't like water. Now, again, this is biochemistry. So those of you that, if you like biochemistry, this is biochemistry. So essentially, the hydrophilic head is a charged particle. Anything with a plus or a minus in chemistry can bond with water. Water will be attracted to things that have a positive or a negative charge. So the phosphate group, can anybody remember, and in biology you have to know as well, you find it in DNA as well, the phosphate group. So why do we need phosphate in our diet? Well, we need it because it's in DNA and it's in our cell membranes. So that's why we need phosphorus in our diet, the element. +5, what's that? -8? Yeah, that's right. So PO4 3 minus. So that's charged. The phosphate group means that the head is charged. It's going to be attracted to the hydrogen in the water molecule, right? So it's going to be attracted to hydrogen. The tail is hydrophobic. So yes, the tail is just carbon and hydrogen. If you take away the carboxylic acid bit that's bonded to the alcohol, to the glycerol, which is up here somewhere, the carboxylic acid, that's kind of not charged. It's all locked into a bond. The rest of the tail is just carbon and hydrogen atoms, and they're not very, well, they're not charged. So, yeah. So they repel water, absolutely. Hydrophobic. The tails hate water. Now, what this means is that our cell membrane arrange themselves like this. If you chuck some phospholipids in there, into our body fluids, this is how they're going to arrange themselves. And you could think of this as the inside of the cell, so cytoplasm. Now the cytoplasm is going to mainly be water, H2O. I know it's got sugars, it's got enzymes, it's got proteins, it's got glycogen molecule, it's got all sorts of things dissolved in there, but the cytoplasm is mainly water, so the heads are going to be attracted to the water. And then what is outside the cell? Well, your cells are bathed in tissue fluid, so there is a liquid around your cells. Of course, we're not insects, so we don't have blood. Our blood is in a closed circulatory system. It's in our veins, arteries, capillaries, venules, arterioles. It's in the blood vessels. But we do have tissue fluid around our cells, and that's going to mainly be H2O as well. So the heads are attracted to the outside and the inside of our cell. And this is a good thing because this creates a bilayer, 2 layers is what this is, the phospholipid bilayer, 2 layers of phospholipids. And it means that charged particles cannot enter our cell without permission. Okay, good. And you have to be able to explain this. So there are many questions where you're asked 5 marks, like describe how the cell membrane is arranged, describe the arrangement of the phospholipid bilayer or something. And that's where you have to be able to say, well, the phospholipids heads face outwards towards the tissue fluid, and the hydrophilic heads of the second layer face inwards towards the cytoplasm, so they can interact with water with the 2 sets of tails in the middle, hydrophobic layer. And this is a good thing because it stops charged particles. And we just talked earlier in the lesson about why we don't want charged particles entering without permission, because they're going to lower the water potential. Could cause all sorts of problems. So we want some control over what comes in and out. So how do we get control? Yes. That's a really good point. So yes, some organelles are contained. You have membrane bound organelles. Now this is one of the things when you talk about prokaryotic and eukaryotic cells. When we talk about prokaryotes like bacteria, they don't have membrane bound organelles. What's a similar thing they have? They have cytoplasm, but that's not really an organelle. What else do bacteria have? They have ribosomes, but ribosomes do not have a membrane. Ribosomes are made of protein and rRNA. But things like Golgi apparatus, mitochondria, chloroplasts, you can't find those in a bacteria. They are double membrane bound organelles. They have a membrane. Yeah. Anyway. Yeah, they have a membrane made of phospholipids. Oh yeah, let's have a look at that. So yeah, maybe not all of the organelles. I can't remember if a vesicle is a vesicle is bi. It has a bilayer or a single layer. Anywho, it has phospholipids around it because vesicles. Remember, vesicles carry substances which could endanger the cells, like enzymes or something. They can carry them and release them outside of the cell by exocytosis. Now they need to be made of lipids as well, phospholipids, because they're going to blend in and become one with the cell. So that they then, and then when you have things budding off, when you do endocytosis and something enters the cell, you can have a whole piece of the membrane can bud off and form a vesicle. Anywho, so how do we have some structures inside the cell, guys? So what do we call something like this? What are these things doing in the membrane? Anyone got an idea? Oh, let's make a little funky. Okay, let's not go too funky. No. If I had the art skills, I would draw little hexagons here for this, but I don't have the art skills for that. Okay, so you have to allow me there. Yeah, we got a channel protein. So this one here is going to be a channel protein, and some of these things can move and some of them don't move. It was called the fluid mosaic model because basically these phospholipids are not static, they're moving around each other like this, rotating, and these channel proteins, some of these proteins can move as well about like that. They can wander about, and others are actually scaffolded in, structured in, and they can't move. They're locked into place. This is a channel protein, and does anyone know what channel protein's for? Someone said intrinsic protein. Nice, very good. We can call this an intrinsic protein or an integral protein. There's quite a lot of names, as you know, in biology. Biology's like that. You got, like, think about sex cells. You call sex cells, gametes, whatever. There's like so many different words in biology for the same thing. So this could be an integral protein, intrinsic protein. Then you've got extrinsic. This could be called an extrinsic, I can't even pronounce it, or peripheral protein, meaning it's on the outside only or only on the exterior. So only sticking out one side of the cell. We could call that an extrinsic protein. Channel proteins are someone said active transport, not used in active transport. So actually that's a good thing. This is why it's good to have the chat and people making comments, because you can clear up misconceptions. So only used in facilitated diffusion. Yeah, we don't have active channels, so we have facilitated diffusion. Now we can have gates on certain channels. You can have gates that only open at certain times. So when you do the nervous system, you'll learn about gated channels, but they will only do facilitated diffusion channels. Once they're open, they're going to allow certain molecules in, like amino acids and things, to just diffuse through here down a concentration gradient into the cell. We do have, does anyone know the names of proteins that do active transport? No, facilitated diffusion is not regular diffusion. I will come to that as well. It's a really good point. So intrinsic protein only used in facilitated diffusion. So what type of molecules diffuse? So for polar molecules or large molecules, really large molecules like enzymes and things need to enter by a different mechanism called endocytosis, which is where the whole membrane dips in like this, and the enzyme or the big large molecule, which will be made of like a polymer, a really big polymer, will come and it will bud around it, and then it will pinch off and break off, and it will be surrounded by phospholipids, and it will travel as a vesicle into the cell. Okay, so that is endocytosis for really large molecules, but quite large molecules and polar molecules means molecules which are positively or negatively charged. Think of amino acids, they have an amine group, nitrogen and hydrogen, so that's going to be charged, and they have oxygen and they have a carboxylic acid group at the other end. So amino acids are charged molecules. They cannot come through the cell like this. Small non-polar molecules can go through like that, and they have to be fat soluble. So you're looking at covalent molecules generally. So things like carbon dioxide and oxygen can just go in and out down a concentration gradient. Generally CO2's going the other way, yeah? It's not really going to be diffusing into the cell. So small non-polar can, I'm just simple diffusion. So if you're small and you don't have a charge, you can go in, you're allowed in by simple diffusion. You don't have to wait and go through a channel. Right, let me just look at some of the questiones, because a few questions. So where are we? Is facilitated diffusion just regular diffusion? Yes, it is, from a high concentration to a low concentration, but it's through a protein. And we'll come onto that and the limitations of that. So there's a limitation to that. Which one? What's the limitation to facilitated diffusion? We're moving from high to low, but what would be a limitation of facilitated diffusion? You should think about, like, say I had 1,000 amino acids outside and only one amino acid inside, we're going to get a very high rate of facilitated diffusion. But what if I have 1,000,000 amino acids outside? There is a limiting factor where you won't get a faster rate of diffusion. So it's the amount of channel proteins or carrier proteins. Now, an important point, someone, Mathilde said channel proteins or carrier. There are carrier proteins that do facilitated diffusion as well. But for active transport, they only do carrier proteins. I haven't shown a carrier protein yet. I got this bad boy here. So let me see if anyone can tell me. Okay, someone said co-transport and facilitated diffusion. I will come to that in a sec. What's this type of protein called? Anyone got an idea? This is a special protein, and it doesn't look like it goes all the way through, so it might be a peripheral, probably a peripheral protein, but you never know. Peripheral or extrinsic. So you could call this extrinsic, means it doesn't go all the way through. You might say, well, what's the purpose of that? Well, we'll come to that. That is a glycoprotein. Well done. Awesomeness. Good. Glycoprotein. And, okay, give me 3 functions of glycoproteins. Now, whether you're OCR, EdXL, or AQA, you need to know at least 3 functions of glycoproteins. Okay, in the ideal world, I'll be able to draw little hexagons to show you that it's made of glucose molecules, i.e. glycogen. It's a polymer of glucose, heavily branched, obviously. Good. Cell recognition. So, do you remember in GCSE when you did vaccinations and you did immunity in biology? You may have even studied immunity at the moment. What do we call the proteins that are on the outside of every living organism which are unique to that organism? Yeah, cell recognition, nice. Do you remember the ones that antibodies hunt down and destroy? Not antibodies, although you are right, antibodies can be glycoproteins, but, well, they're not glycoproteins. They're antigens. So cell recognition, they can play a role as an antigen where they're recognized by a specific 3D shape. They could be a marker. What do we mean by cell signaling? Well, certain hormones, you all did I'm thinking about GCSE because some of you might be in year 12. So in GCSE, you will have done the whole mechanism of insulin. So, like, how does insulin go to your liver cells and say, open up, you need to put glucose in because blood glucose has got high? Well, it binds to a glycoprotein, something like that, so, and causes conformational or shape changes on the inside that maybe open a carrier protein or something like that, or trigger something. So there's loads of these. You've got hormones going back and forth between cells. They hit a cell and they can cause a 3-dimensional change of shape of a protein on the inside, and you get a cascade of reactions going on. So, but you just need to know glycoproteins are used for cell signaling, cell recognition. Another one is cell adhesion. So they can use them like scaffold. So when your cells, how do they hold on to the cell next to them? So they can attach to cells next to them and they form tissues. How do we get skin tissue? How do we get muscle tissue? Well, these glycoproteins lock on to the cell next to them, so the cells stay in a structure basically. So they can be for cell adhesion, which means sticking cells together, cell signaling, which means receiving hormones, so they're part of the communication, cell communication. And then we've got cell recognition, so your white blood cells know if it's a foreign invading cell or if it's a self cell. Anywho, yeah, receptors, that's right, they can act as receptors. Cool. What else? Right, Mathilde, plasmodesmata in plant cells are gaps in the cell wall, not the membrane. So the plasmodesmata are little holes in the cell wall that allow water and things that are soluble in the water to move between adjacent plant cells, but not in they're not holes in the cell membrane, they are holes in the cell wall. And they're in plant cells only, the plasmodesmata. You don't get those in animal cells. But they so yeah, little gaps between the cell in the cell walls of adjacent plant cells so that water can move or substances can move. What else? Oh, one other thing to recognize before we move on off of this. What do you think I would call this molecule? Oh my days, what is that? What would that be called? It'd have a slightly different name than this one, so watch out for that. Now at A level, they don't really focus on the functions of this much at all. Glycolipid, nice. Notice it's attached to a lipid, one of the phospholipid heads. So we call this a glycolipid. And you kind of like the functions are all the same. You could Google it and find out what unique functions they tend to have, but glycolipids we tend to just bag them in with the same kind of thing as glycoproteins. You don't tend to be asked specifically about glycolipids, but you may be asked to name it and be expected to know the different name of it. Right, I'm just going to do a quick crusty little diagram. All of my days, check out this awesome graph. So I just want to show you before we move on to I haven't really talked about active transport much, so, and then someone mentioned co-transport, so I'll talk about that in a second as well. Co-transport is slightly different than, it's similar to facilitated diffusion, and you're absolutely right, it involves protein channels, but it also co-transport. It just means you need two things to go through, and some of the mechanisms of co-transport are complicated and some are simple. Yeah, co-transport occurs when you do active loading of sucrose from companion cells into the companion cells, basically during translocation. And it also occurs in the small intestine co-transport with how glucose is moved into the blood. Now from the lumen of the small intestine. Right, we want rate of transport across membrane, right? So you could have a graph like this, rate of transport across membrane, and we could have external concentration of substance. I don't know why I wrote outside the cell. If I've said it's the external concentration, it's obviously outside the cell. I'm just trying to make you aware that, like, you could have something like this. Let's just do that. And then I could have a second line. Oh my days, why is that so fat? I could have a second. Oh yeah, let's not try that, Tom. Let's just go with a blue line. Oh my days, what have I done? Yeah. So No, that doesn't work. I wanted a smooth line. Never mind. Oh, that's crusty, isn't it, really? So, what mechanism of transport do you think A is, and what mechanism do you think B is? This was a crusty diagram. Um, yeah, very good. So, basically, I think Mathilde has pointed it out here. The rate of transport across the membrane, one of them just keeps going and keeps going and keeps going, and one of them after a while plateaus. And so A is simple diffusion and B would be facilitated diffusion. So after a while all of the channel proteins are busy. So if you have 500 channel proteins in your membrane, then you have a limit of, I don't know, let's say it takes 1 second for a molecule to move through that protein, that channel protein. Then you could the maximum intake of substances into your cell is 500 amino acids a second. Now, it doesn't matter if I then say, well, increase the external concentration to 5 million. Diffusion is from high to low, and the steeper the concentration gradient, the faster it moves. You're not going to get any faster because they're queuing up to go through the channel proteins. So you can say the channel proteins are saturated, or like, but essentially it doesn't matter if we keep increasing the external concentration, you will not get faster rate of diffusion. Whereas with the lipid membrane, the lipid bit, so things like oxygen, if you keep increasing the concentration, the oxygen will just move across faster because you've got the entire membrane to get across. Yeah. And active transport would be up here somewhere, just a straight line across. It would not matter if you're doing active transport, changing the concentration doesn't really affect it. It's just going to be more or less a straight line across the top here. So you're going to have a very high rate of transport, and it won't matter how many are out. It doesn't matter whether it's a low or high concentration. It's just plugging away every second, moving the same amount across, whatever amount that is. It's going to be moving the same amount across every second. So active transport, we move against a low concentration to a high concentration, and it obviously requires energy from ATP, which is normally formed in aerobic respiration. Yeah, we've talked about glycoproteins, glycolipids a bit, facilitated diffusion. So just remember with facilitated diffusion, you are still going from high to low. So obviously, if I increase the amount of particles here, they're going to move faster. I'm making a steeper concentration gradient. So that is facilitated diffusion, and that is charged particles. What else? What else? Simple diffusion, we already talked about. That's just like oxygen. More than likely, carbon dioxide's going this way, out of a cell. You're not going to have carbon dioxide going into a cell. Cells produce carbon dioxide by respiration, aerobic respiration, and so it's generally always going to be oxygen in, carbon dioxide out, and they just go straight through the membrane. What about water? Does anyone know how water gets in and out of cells? What's the process by which water moves in and out? Oops. So water is going to move in and out. Oh my days. By osmosis, and sometimes through things called aquaporins, which you might not have covered yet, but things called aquaporins. And aquaporins are just special protein channels specially for water. And actually we can increase the number of we can actually add these proteins. So if it's a really hot day, you can add more aquaporins to the cells in your kidney, in the tubules, and you can reabsorb more water, so you wee out less water. And that's what happens when your urine is very dark yellow. You've released antidiuretic hormone from your pituitary gland, and it has caused more aquaporins to move into the cell membrane of your collecting duct and your distal convoluted tubule in your kidneys, and it means more water can be reabsorbed. So we have these special proteins channels, and when it's for water we have a special name for them. They're called aquaporins, and that's where osmosis happens through. Cool. And it wouldn't be called facilitated diffusion, it would be called osmosis. Superbulous. Now, what else about the membrane? Let me give you a couple of questions while Hormadace. Now, I didn't really talk about 5. I think I've only mentioned 3. So, see how many you can reel off. You can just name, you don't have to describe, but see if you can name then. Oh yeah, haven't mentioned cholesterol. Thanks. Let's go away from this question. Boo to that question. Let's go back and smash cholesterol in. So, see these little red lumps here? It's my incredible drawing. I know. Van Gogh in the house. These are cholesterol. What is the role of cholesterol? Does anyone know what cholesterol does? We need cholesterol. Without cholesterol, we all thought cholesterol was bad. Well, it's not bad if it's the cholesterol that goes into your cell membrane. It's bad if it's the low density lipoproteins that cause lipids to move out of your liver into your blood and increase your chance of atherosclerosis, fatty deposits inside the artery walls. That's bad. But this cholesterol's good. This cholesterol plays an integral role inside amongst the phospholipids. It is a lipid. Cholesterol is a lipid. So it's a non-polar molecule. Yes, it regulates fluidity. It actually makes the cell membrane more rigid, more stiff. So makes cell membrane stiffer, more rigid, or regulates flexibility. Now, yes, it plays a role in flexibility by making it stiffer. Without the cholesterol, the phospholipids will wander off and all the cytoplasm will leak out into the tissue fluid and the cell will collapse. So without the cholesterol, the cell membrane will collapse, and especially in animal cells which don't have a cell wall, that would be disastrous. Imagine things like your red blood cells, which are they don't do cell adhesion. So red blood cells, it'd be a disaster if they started doing cell adhesion and sticking to other red blood cells. You get blood clots. We don't want that. So they have to be free single cells flowing through your blood vessels, and we don't want their cell membranes falling apart. So cholesterol makes the cell membrane stiffer, and that's what you have to know. Good, it maintains fluidity. I haven't talked about what can disrupt the cell membrane. So do you guys know some things that can make it more permeable and allow more stuff to get across? What kind of factors do you think would increase the speed at which amino acids or glucose or salts, sodium ions or things move across? What would increase the speed? More channel proteins, absolutely. If you increase temperature. So the hotter things are, if we make our oxygen particles have more heat, they have more kinetic energy, move faster, faster rate of diffusion, simple. Faster rate of simple diffusion. If so, increasing temperature increases the rate of diffusion, and that whether it's facilitated or simple diffusion, both will go faster at higher temperatures. What's going to happen at really high temperatures? What do you think would happen to fats and oils at really high temperature? What if you put some some butter in a frying pan and you warm it up? What happens? What might happen to our lipids at very high temperatures? And then if you've studied enzymes, which you did at GCSE, well, the lipids won't denature, but these protein channels will denature. The lipids will melt. They'll literally melt. So they are made of the same stuff as butter, they're fats, they're lipids. They're going to melt at high temperature and become liquid. And that is going to mean all the contents inside the cell just spill out. And there's a practical you do with this where you use a red substance, a pigment, inside beetroot cells, and you vary the temperature and you use a colorimeter, and basically you're measuring absorbance, or you can measure transmission through the solution, but when you heat it up, the membrane melts and the red pigment just flows out the cell into the solution. And obviously, if you're measuring absorbance, it's going to absorb more light, or if you're measuring transmission, you're going to get a decrease in transmission, depending on which one you're measuring to gauge your permeability of the cell. So essentially, also, good point, someone said, you're going to melt these, you're going to denature, which is the key word, proteins, and that's going to make things move in and out really easily from the cell as well. Yeah, so you're doing that practical next week. Yeah, so essentially you might be investigating temperature, or you might be changing you might be adding solvents. There are also things like alcohol that will dissolve fats. There are certain detergents and certain things that will, certain detergents, I'm not sure, but certain solvents anyway, like alcohols and things, will dissolve the cell membrane, and that obviously make it more permeable, but it will rupture or damage the cell. You can just say damage the cell membrane and increase its permeability. So yeah, denaturing proteins channels is one of those things, and melting the phospholipids at really high temperatures will unit 1 or 2, it depends which course you're doing. So I just kind of waffled about a lot of stuff to do with transport across the membranes. There is something called co-transport, and co-transport, there are lots of different ways of co-transport. Co-transport tends to be when the word co, two things cooperate to cross the membrane. So sometimes certain ions or certain molecules cannot cross the membrane unless they're in the presence of another ion, and they'll go together through a facilitated diffusion mechanism through a protein channel. Now, I haven't also sodium in the ileum. Yeah, so hold on, I've got, where's that? I can just put up the CGP on that, one sec. Doesn't it overlap between both? Unit 1 and 2, I can't remember. It depends which exam board you guys are doing. Oh, talking of the ileum, all right, you don't have to need to read all of this. So the mammalian ileum, this could be a rat, this could be a dog, it could be a human. A human? Never heard of one of those. But glucose is absorbed by co-transport in the mammalian ileum. And I'm better off drawing my own diagram rather than using this one because it looks a bit poop. But essentially, what happens, these cells here are the epithelial cells. It's better if I draw it myself, but I'm a bit lazy and I didn't do it earlier, so there we go. The lumen is the inside. Now remember, you on both sides of the lumen, this side and this side, you would have epithelial cells, well, going all the way around like that. So the food is moving down the middle, and if we just we basically want to encourage the uptake of glucose. And the way that glucose is moved in, it's moved in through a co-transporter protein with sodium ions. So you need to have some salt. Now, you don't need to add any salt in your diet. There's salt in vegetables, and all foods naturally have sodium ions in them. But if you had no sodium in your diet, you'd be in big trouble because you wouldn't be able to absorb glucose into your blood. Your brain cells wouldn't be able to respire. You'd basically be dead. So you need sodium in your diet for the absorption of glucose. And what happens is, a pump here at number 1, this is a sodium potassium pump. It takes sodium ions and it pumps them out of the epithelial cells into the blood capillaries next to the epithelial cells. So the epithelial cells is the layer of cells on the lumen, the single layer of cells on the lumen of your small intestine. So the food's there, the epithelial cells are here. They pump sodium into the blood, which is taken away. And this creates a concentration gradient between the sodium that you've eaten in your lumen, and it's very low in sodium in the epithelial cells. So then sodium says, hey, I want to come in. So sodium moves down through a type of facilitated diffusion through a transport protein called a co-transporter. As the sodium moves down, the sodium, because it's positively charged, binds to a glucose molecule and drags the glucose with it. And so you might say, well, why does it do that? And then the glucose can just move by facilitated diffusion into the blood. Why can it go by facilitated diffusion once it's in here, because the blood is carrying away the glucose and maintaining a concentration gradient where the glucose constantly wants to move across. So we might say, sir, why do we need sodium ions to drag the glucose through? Why don't we just let the glucose move through? Why don't we just have a facilitated diffusion channel? What's the problem with diffusion? The problem with diffusion is you can never, like, you're going to struggle to get more than 50%. Say I've eaten a burger or whatever, I don't know, a chocolate bar is full of glucose, and I'm going to struggle to get more than 50%. As the sugar's coming down here, it's diffusing across. Once this cell has 50% glucose and the lumen has 50%, no more glucose is going to move across, right? It's not going to move across because it goes from high to low. So this mechanism of pumping sodium ions out here, actively using energy, using some ATP to pump the sodium into our blood, means low sodium here. So sodium moves in and carries the glucose with it. So the glucose is kind of moving against the concentration gradient. It's a form of, they don't use this term anymore if you're doing AQA or OCR. They don't use this term, but it's a form of indirect active transport. We're actively transporting sodium ions to create a concentration gradient for the sodium. The sodium moves in and drags the glucose with it. The glucose is moving against its concentration gradient, but the glucose isn't being actively transported. It's actually diffusing against its concentration gradient because, I know, how can you get diffusion against the concentration gradient? Because the sodium is diffusing in and dragging the glucose with it. Anywho, complicated. What you really just need to know is that's called co-transport. Sodium and glucose move by co-transport in the ileum, but that's a little bit complex. Yeah, so the potassium is pumped in. This pump is in your nervous system as well. It works by pumping 3 sodium one way and 2 potassium the other way. That's just the way that pump works. Yeah, so glucose then diffuses out into the blood by facilitated diffusion, which is fine because the blood is rushing past here, maintaining a concentration gradient. So you're not going to have to worry about actively transporting glucose in here. This is kind of like us cheating to actively transport the glucose in here because we want to get all of the glucose in. We don't want to like poo out or excrete out the glucose when it can be used for respiration. So this is a way of getting the glucose to go against its concentration gradient. Indirectly active transport, you're doing active transport of sodium to create a concentration gradient. Anyway, that's the co-transport of sodium. You can read up on that if that is the kind of thing you like. Let's do a few questions on the membrane. No, the plants can receive substances. Plant cells can still receive substances even though they've got a cell wall. See if you can, what 2 answers would you go for here, guys? So Gala's going A, Matilda's going A and C. So which statement describes a feature of plasma membranes? Channel proteins are a type of intrinsic protein. That sounds pretty good. Glycoproteins are intrinsic proteins that have lipid. See, that doesn't make sense, does it, B? If you're glycolipid, you're not a protein. So that's wrong. Phospholipids form a rigid bilayer. No, it's fluid, hence the name fluid mosaic. The plasma membrane forms an impermeable barrier. No, it's permeable. All parts of it are permeable to something or another. So yes, the answer is A for the first one. The second one, how can molecule X be described? Hydrophilic head, hydrophobic tail. It is formed when the glycerol in a triglyceride is replaced by a phosphate. Is the glycerol replaced? It's not. Glycerol is still, so B's wrong. You still have a glycerol, you just have a phosphate group added to it. It is non-polar. What is X pointing at? It's pointing at the head. So the head is not non-polar. The head is polar, is hydrophilic, which means polar. The tails are joined to the head by peptide bonds. Both are A. Both are A. Both A. Yeah. The peptide bonds, where do we find peptide bonds? We're not going to find them in the membrane. We're going to be dealing with ester bonds in the membrane, which join the glycerol to the fatty acid tails. We're not going to... Oh, sorry, you will find peptide bonds. You'll find them in the protein channels. Yeah, sorry. You find them between the amino acids in the proteins, in the various proteins, but you won't find them in a phospholipid. Cool. He said something stupid there. Okay. Let's do a couple more of these. I haven't talked about exocytosis and endocytosis. They're not that big on that in the A-level syllabus anymore. Endocytosis and exocytosis. We're not too big on that in AQA or OCR too much. Exocytosis, exit, means leaving the cell, and endocytosis means coming into the cell. And the way this works is substances, this is something leaving the cell. We can guess what that is, exocytosis. So imagine this grey part is a layer of phospholipids. They're surrounding a substance like enzymes or something they want to leave the cell. It joins and becomes part of the membrane. So this fluid membrane, these vehicles come and then they form part of the membrane and just release their contents outside. And the opposite way round, if this was going the opposite way, these substances would approach the cell and the cell would dip like this, and it would you just turn the arrows around the other way. It would do that, and then it would pinch off and go into the cell, and that would be endocytosis. Go. Endo takes in, exo goes out. You guys are spot on. Diagram below shows the structure of a plasma membrane. Which label A to D indicates the component of the membrane that can affect its fluidity? Oh my gosh, my eyes can't pick that up. Yeah. Well done. I think you're right. Yeah, you're right. So if it was right. So Mathilde, yeah, phagocytosis is very similar to endocytosis. It's very, very similar. It would do the same thing, but it would form a slightly larger thing, and it's a vesicle. So the same thing. You're absolutely right. A phagocytic vesicle. Some exam boards call it a phagosome. Some exam boards call it like a vacuole. And this is the... Do you remember at GCSE we used to say animal cells don't have a vacuole? Plant cells have a vacuole. So at A-level, technically, you should say plant cells can have a permanent vacuole, animal cells don't. White blood cells are an animal cell and they can have temporary vacuoles. Say they're taking in some bacteria, they will form, similar to this, they will form a phospholipid layer around the bacteria that we call a phagocytic or phagocytic vesicle, but it's a kind of vacuole. So yeah, white blood cells can form a temporary vacuole. So that's why we use the word permanent when we say plant cells have a permanent vacuole. Some animal cells can form vacuoles when engulfing or eating, like consuming stuff. Anywho, yeah, you got that right. So yeah, it was D, the cholesterol there, a bit hard to see. Let's have a look at some other questions. These questions are from all different exam boards, OCR, EdXL, AQA. What else? Someone, grab that. Yeah. So someone said about plant cells. I saw there's a comment. How do plant cells receive stuff? They got a cell wall, and someone answered in the chat the correct thing. Plant cells, cell walls are permeable. So they're permeable and they have little holes. When you have plant cells, you sometimes have little holes. Say this is one plant cell. Oh my god, this is the most crusty drawing. Yeah, that's a very crusty drawing. So two plant cells all next to each other. There are little holes like this, but of course, that doesn't help with external transport. Those little holes, I've not made much of a channel. That's plasmodesmata. So stuff can flow from the cytoplasm here, there, and then into the next one, and then into the next. But, yeah, the cell wall has to be permeable to allow things through it. So the cell wall is permeable. D, right. D. What we going? B, D, B, D, B, B, D, B, D, B, D, D. Yeah, I think, let's check them out. Glucose by active transport. No, that's simple diffusion. That's going to be simple diffusion. Right, the reason, so it can't be, it's not active transport, we've got simple diffusion. The reason it can't be B, is because glucose is charged, and charged polar molecules cannot cross the tails, the phospholipid tails. So we can get rid of B as well. Then we'll look at C, oxygen by active transport. No, that's going high. Look, 4 there, 2 there. It's going from high to low. It's diffusing. And active transport would be through a protein. I still haven't shown you a picture of active transport, which is bad. I'll put that up in a sec. So yeah, so it's going to be oxygen by diffusion there. And this is glucose by facilitated diffusion. Notice they just say diffusion, but it's actually facilitated diffusion. So both are diffusion. This is just, so this must be glucose through a protein. Okay, cool. Superbulous. Right, I'm going to look at active transport after this. Which of these could contain a sulfur atom? 1, 2, or 3? I can't really see what is what. So, what you got to say to yourself is, what is number 1 pointing to? It's pointing to a protein. Number 2 is pointing to like a glycolipid, but essentially the glycogen part. And you think carbohydrates, they contain carbon, hydrogen, oxygen. And then we go, this is pointing to the phosphate head, which is going to be, lipids are carbon, hydrogen, oxygen. Phospholipids have phosphate, 4 elements. And then proteins can have those disulfide bridges, right? So some of the R groups could contain a sulfur atom. So the answer is going to be D for this one. 2 is a glycogen, a glucose molecule, and even if it was contained to the lipids like carbohydrates are just carbon, hydrogen, and oxygen. Proteins are carbon, hydrogen, oxygen, nitrogen, always, and sometimes sulfur. And once they're formed later on, they can actually take phosphates on board in certain proteins, but that's not naturally like that. That's later on they can do that. You only need to know for A level carbon, hydrogen, oxygen, nitrogen, and sulfur for proteins, 5 elements. Okay, let me zing just quickly to do active transport. Is that active transport? Right. So again, I've rather than draw my own diagram, I've kind of teethed that. Oh, good. Hold on, Gala. So the protein will be the big fat thing. Then it's a glycoprotein. If it's a lipid molecule, it will be attached to one of the heads, the phospholipid heads. So, rather than a protein. So it's not, that is still glycogen. That was, oh, where's that question gone? I've lost the question. Okay. This is glycogen. This is glycogen. And this is glycogen. But it's what they're attached to. So these guys are attached to, it looks like, I can't really see with my eyes. I've got bad eyesight and this doesn't come out very clearly. But this looks like it's attached to the phospholipid heads. The big fat things in the membrane, the biggest molecules, are the proteins. So when the glycogen is attached there, it's a glycoprotein. When the glycogen is attached here, it's a glycolipid. We don't say glycophospholipid, we just call it glycolipid. Anywho, let me zing back to the old active. Right, active transport, 2 mechanisms. This is a carrier protein. All right, it's you don't have to read all of that, guys. Active transport is the movement of substances. You did this at GCSE from a low concentration to a high concentration using ATP, using energy from respiration. At A level, we use the word ATP. and ATP is hydrolyzed, which means broken. Whenever you break a biological molecule, it's hydrolysis, and you use water to do that, and it turns into ADP, adenosine diphosphate, and inorganic phosphate, and it releases a little bit of energy, and that little bit of energy will cause the shape of this channel protein to change and release the molecules onto the other side. In this way, it's calcium. So it's saying there's a low concentration of calcium. We've got 1 here and we've got 3 inside the cell. This 1 calcium has gone in here, and essentially ATP has been used to move this across, and you'll kind of get ATP going to ADP plus PI. And so active transport only uses carrier proteins, not channel proteins. There is a method of facilitated diffusion that can use carrier proteins, but you rarely ever have to specifically know that. And all it is that it's passive. There's no net use of ATP. So even if ATP is used on one side, it's created on the other side, but they don't go into that detail anymore. You just have to know that facilitated diffusion is passive, and when we say passive, it means it doesn't, there's no net overall use of ATP. Anywho, that's a carrier protein, which is for active transport. Yeah, I'm not sure what it was attached to there. And then let's have a look at a couple more questions. Did we do this one? We did this one. Yeah, we did that. Let's have a look at. Okay, let's have a look at this question. Okay, so I've got a couple of questions here on the membraner. The diagram shows the structure of a cell surface membrane. The molecule R is a phospholipid. Yes. Give the name of the bond that joins a fatty acid molecule to a glycerol in a phospholipid. Yep. The bond in phospholipids and that you have to know about is a ester bond. Boom! Yeah. Nice. And what you're going with? Yeah, ester bond, and you're going with C and A. Let me check that out. CAT you're going with. Right, let me check. Which of the following describes molecule P? So that is a glycogen attached to a lipid. So we're going with a glycolipid. And this was ester. Everyone go for C for that first one. This is a glycolipid. That white thing there, that's the hydrophilic head of the phospholipid, and then these are glycogen. These are glucose molecules, which are probably, well, yeah, they're forming glycogen as animals don't have starch. Which of the following molecule, not that it says it's plant or animal, so which of the following molecule is involved in cell recognition? Is it Q? So for recognizing the cell, is it Q? Is it R? Is it S? That looks like cholesterol. Or T, which is the facilitated diffusion channel? It's going to be Q, which is A. So the answer is A for this one. So give yourself a pat if you got that right. This glycoproteins are used in cell recognition, like as antigens. And then, yeah, that's right, Q. Which molecule allows ions to diffuse into a cell? Of course, that is going to be facilitated diffusion, and that is going to be D, that one there. Ions are charged, so things like calcium or sodium, they're not allowed to go through here because they're not soluble in this the hydrophobic tails, so they can't cross the bilayer. Yeah, sodium, potassium. Now, sodium can go by facilitated diffusion, and you have pumps. Now, pump, Think of the word pump. Let's go pump some weights. All right. Think of the word pump. It uses energy, pumping something, pumping up your bike tire. That looks dodgy, but pumping up your tire uses energy, and so it's active. So sodium potassium pump is active transport. Pumps are always active. They use energy, but sodium doesn't just come. It isn't just transported by active transport. It can enter by specialized channels that will let them in, sodium channels. And your whole nervous system relies on the fact that there are facilitated diffusion sodium channels, ion channels, and there is a sodium potassium pump actively transporting sodium. That's how electrical signals or impulses are sent around your body. Without sodium, and potassium, your nervous system would not work. Yes, your sodium and potassium pump. Right, couple more questions before we finish up, because this was just a quick revision over this. Okay, this is an edXL one, so if you're an edXL person, I don't think any of you guys are doing edXL, but if you're doing edXL, this is an edXL question. In the lungs, oxygen moves from the alveoli into the red blood cells. How many times does an oxygen molecule cross a cell surface membrane to move from the center of an alveoli to the center of a red blood cell? So you've either got to visualize this in your head or draw an alveoli and then draw how it gets into a red blood cell. That one's quite hard, I reckon. Oxygen enters the red blood cell and then you have a choice. If I draw it for you, you'll get it straight away. So here's your, oh my days. All right, this is some crusty drawing, but this is the alveoli. Does anyone know the name of the cells that line the alveoli, guys? What cells line the alveoli? You have to know this for all your exam boards. They're squashed, which is kind of, I think of the name as like, it looks like squashed when you write it. Now, cells that cover layers inside your body are called epithelial cells. These are squamous epithelial cells. And if oxygen is here, how many membranes does it have to cross to get out of the alveoli alone? Well, these are cells. These are squamous. They're not plant cells. I know they look a bit brick-like, but they're squashed. Squamous epithelial cells. It's got to cross two membranes just to get out here. Where are we going to find our red blood cell? Is our red blood cell floating past the alveoli? Here's our red blood cell. Is our red blood cell just... Oh, red blood cell's donut shape, but I don't want to draw it like a donut because it will confuse. But what is the red blood cell found in, guys? What's the red blood cell found in? We don't have what we call an open circulatory system. Red blood cells are not allowed just to float. When you get a bruise on your arm, that's when the blood is leaking into your tissue fluid and you're seeing the blood as a bruise, okay? But normally it's kept inside blood vessels. So it's going to be in a capillary. How many cells are in the wall of the capillary? So check this out now. Now you're going to see this question was hard because now you got I think the capillary's got squamous epithelial cells. So there's my capillary, one cell thick, same as the alveoli, one cell thick. Now you've got to work out that oxygen has got to go into there. How many membranes has it crossed? How many membranes has that oxygen crossed? Not how many cells, how many membranes did it cross? I think it's either 4 or 5. Let's check it. It's going to go 1, 2, 3, 4, and then 5 into the red blood cell. So it's 5. It's actually 5. It's got to go through this cell membrane, which is the same cell, cross across the cytoplasm, across that cell membrane, over a third cell membrane into the wall of the capillary, which is made of cells, across the cytoplasm, out of that cell membrane, and then across the cell membrane and into the hemoglobin in the red blood cells. So it's actually 5. That was a hard question. 5 cell membranes. Um, do red blood cells have a one cell thick membrane? No. So Matilde, that's a really good question you're asking. So a red blood cell just has a bilayer like your other cells, a phospholipid bilayer. Um, it is only one cell, so that's a misconception. It's only made of one cell, so its membrane is not going to be one cell thick. It's not as thick as the whole cell. What we say about the capillary is really a tissue, so that's a wall. Remember, the other misconception that people will do, they'll call this a cell wall. Animals do not have cell walls. So we'll talk about the wall of the capillary is one cell thick, but don't say, oh, the capillary has thin cell walls. It can't have a cell wall. It's an animal cell. So the capillary is actually a tissue. Walls of capillary, one cell thick. So the walls of the capillary are a tissue. They're a group of similar cells doing the same job, but it just means they're one layer thick, just like the epithelial cells in your small intestine, like the not ciliated, the ones with villi, the villus are one cell thick as well, the walls of the small intestine. But the red blood cell just has a thin membrane, I guess. But yeah, their cell membrane. Oh, and talking about the cell membrane, what I didn't do is talk about Fick's law in terms of cell membranes. So cell membranes can have different surface areas. For instance, in the small intestine, the villi create a large surface area for those cell membranes across there, and that will increase the rate of diffusion. So, and the thickness of the cell membrane, you could say as well, but I'm not sure how much variation there is in the thickness. The bilayer is limited by the length of phospholipids. Maybe certain cells have evolved over millions of years if they're on exchange surfaces. I suppose the phospholipids can be different lengths dependent on the length of the tails. So you could have a thinner cell membrane bilayer if the two layers have very short tails with not many carbon atoms in. So yeah, I guess you can have thinner and thicker cell membranes because phospholipids aren't all the same length. It's dependent on the length of the fatty acid tail. So yeah, you could have different thicknesses of cell membrane. But whether or not the red blood cell might have a thin cell membrane. So the red blood cell is just one cell itself, yes. Whereas the capillary is like one of these cells is going to be roughly the size of one of those red blood cells, yeah. B, diffusion. Absolutely, yeah. Oxygen can just enter by diffusion. It's not limited at all. Yeah. I would say generally EdXL is easier. Their course is slightly easier, I would say, than AQA, but you're always going to have. Now, it's good to do questions just from your exam board, but also to flex out and do them from another, because what happens every year is a foreign kind of question is asked in the A-level exam. When you do your A-levels, every time you do your A-levels, when I say foreign, I mean like one that's not an AQA style, you'll get two or three in every year and the kids cry about it on social media and stuff. And really, to get you familiar to like responding to, oh my God, this isn't a normal AQA style question. What's this? I've never seen this before. Is to try ones from other exam boards occasionally. Obviously, don't do ones on content that's nothing to do with your content, but that means that you're prepared for like unfamiliar situations. If you just train yourself as an AQA student on AQA, then you're going to like struggle when you get put in unfamiliar places. Anywho, I think we're going to wrap up there, guys. If you have any questions, you can leave them in the comments. Other membrane, because I didn't get to chat about everything to do with membranes, but if you have any questions about membranes that didn't get answered, you can leave them in the comments. All right, guys. Normally I'm going to be 7 o'clock on Thursdays, so yeah. Normally 7 o'clock on Thursdays, but if you have any questions, leave them in the comments. And take care, guys, and keep working hard for your biology A level, and you will smash it. It's all about consistency, all right? It's all about keep going. In the times when your exams aren't there and your mocks aren't there, you need to keep that work level up, and it's all about consistency. That's what A levels is, all graft and hard work. All right, nice one, guys. Take care, and I will see you next time, next Thursday at 7 for biology, and Mondays at 7 for chem.
I've had training.
The training is nothing. The will is everything. The will to act.
Yield.
You're still here? It's over.